Mm. Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I am your biology educator. Good evening all of you. So all ready? Quickly let me know. Good evening. How eagerly were you waiting for this session? For the revision of Unit 2. 3 more chapters, Morphology, Anatomy and Animal Tissues. Good evening all of you. And please let me know how many of you have tried solving the questions from the Unit 1 and you were able to solve each and every question. Good evening. Quickly let me know. Ma'am, notes of last class have already been uploaded in the Telegram channel. So please, you can find your notes in the Telegram channel. Those of you who haven't joined it yet, bad thing. You have to join the Telegram channel for all the notes and all the notifications, right? And also, guys, before we proceed with the session, I have to tell you two very, very important things. Number one, 19th of April, from Sunday onwards, we are going to start with the mock test series, right? We are going to start with the mock test series, and you have to give those test series, and the timings will be exactly same as NEET 2 to 5 p.m. Also, guys, on Saturday and Sunday, I'm going to complete your 11th and 12th biology. So on Sundays, I'm going to take marathons. On Saturday, we are going to complete class 11 biology, read NCERT completely, underline all the PYQs, highlight all those things. So we are going to read NCERT class 11 on Saturday. Timing would be 7 p.m. So buckle up, right? Because I know before that you will be attending some other marathons also for different subjects. So my time is 7 onwards, no upper limit. So we'll keep going on till our class 11 biology is done. And on Sunday, we are going to complete our class 12 biology. All right, noted. So you will see all those notifications in the Telegram channel. You will get the notes of this session also in the Telegram channel. So don't forget to join. Good evening, good evening, good evening. Quickly share the session with all your friends, call them in, and we are going to start quickly with unit-wise final revision of Unit 2. We are going to cover up 3 chapters in this: Morphology, Anatomy, and Animal Tissues. All ready? How when we cover the unit from second year? Bindu. Unacademy NEET English channel, Telegram channel. The description, it's given in the description box, okay? All right, quickly. Give me a thumbs up. Give me a thumbs up that you're all ready with your note making things. And you know the drill, right? We are going to write down the quick things ourselves. Of course, you will get these notes as well, but you are going to write down them on your own also. You'll be writing down the keywords on your own, just like we did in the previous class. Just like we did in the previous class. All set? All set? Please let me know. Okay. Today only you have joined, so make sure that you subscribe to the channel. Also join the Telegram channel where you will be getting all the notifications, right? Okay, let's get started. Give me a thumbs up that you're all ready. We can start with the session quickly. Quickly. Marathons also. Yes, marathons are planned for Saturday and Sunday. You know, weekend should be special. So we're going to make it special by completing our full syllabus, full biology, one on Saturday, the 12th on Sunday. See, unit-wise revisions will go on, but I'm going to read complete NCERT on Saturday and Sunday with you, right? All right, guys. All right, notes have already been uploaded. Now let's get to the topic. So we are going to start with morphology first of all. This chapter got so much weightage last year. 4 questions from this chapter. So we don't know how many questions are going to come up in this year, but let's buckle up. Let's get ready with with this chapter. So you will be making those points with me quickly. Notes don't have to be nice this time. You will be getting these nicer notes from my side, but you must write them quickly with me. Okay? All right. So starting with the morphology. So morphology is the study of the external body, right? So the plant body is basically going to consist of The plant body consists of what? One, the root system, and second is the shoot system. Okay. The root system is underground. It develops from the radicle, and the function of the root system is anchorage, that means support and absorption of water and minerals. And what is the function? Oh, sorry. What is the shoot system? The aerial part of the plant. That means above the ground. It develops from which part of the seed? It develops from the plumule, right? And what are the functions of shoot system? Conduction, photosynthesis, and reproduction, right? Moving on to the next. Now, starting with the roots. See, roots are of three types. Number one, we have, just a second. Wait a second, wait a second. Okay. Yeah. So types of roots. All right. So types of roots. Wait. All right, types of roots. There are three types of roots, guys. One is the tap root, second is the fibrous root, and the third is the adventitious roots. There is some lag on my side. Wait a second. Just a second, guys. Mm, give me 1 second. So I've already made the notes, but this note fails to work. Give me 1 second. Just stay with me so that I resolve this issue quickly for you. All right. All right. I think it's done. So, yes, types of roots. Number one type of root is the tap root system. Then we have fibrous roots. Then we have adventitious roots. So there are three types of roots. Tap root, which develops directly from the radicle. And the primary root, the one which develops from the radicle, is known as the primary root. And this primary root is going to give rise to the lateral roots. Now, taproot system is a characteristic feature of dicots. For example, the examples, see, there are so many dicots, but the examples which NCERT has particularly mentioned are gram and mustard, right? Then we have fibrous roots. Now, fibrous roots are the ones which are, see, in fibrous roots, the root, primary root, which actually develops from radicle, that is very short-lived. So primary root is short-lived. It is replaced by the adventitious roots which arise from the base of the stem. Are these points clear? This is getting little mischievous. Note is getting little mischievous. Wait. Something is happening. All right. So moving on, guys. Just a second. Guys, please stay with me. I'm so sorry for the inconvenience, but there is some issue. Let me resolve it. The session is very, very important. Yeah. All right. Moving on. Yes, please. So then we have the adventitious root system. Now, adventitious root system is those roots which arise from any part other than the radicle. So one from the radicle is taproot system. The one from any part other than radicle is the adventitious root system. Now, in adventitious root system, we have two types of roots. One is tilt roots and the second is the prop roots or the hanging roots. So guys, be very particular about the examples here. The tilt roots are found in sugarcane. They're found in sugarcane, they're found in monstera, and the hanging roots which develop from the upper parts of the branches, they are found in banyan. Is that clear? They're found in banyan. Moving on to the next now. Sorry. So fibrous roots is a characteristic feature of monocots. Fibrous roots are found in the monocots. Are these points clear? Venation. All right, guys. So are these points clear to everyone? Now moving on to the regions of the root. See, the root is divided into three different regions. See here, the lowermost part, this part, and the upper part. Now this part is known as the zone of maturation. Why? Because this is the region where root hairs will be found. So the region containing root hairs is the zone of maturation. Then this is known as the zone of elongation, and the one which is towards the ground, this is the zone of meristem. So basically, now see the characteristics of each region. The region of maturation is also known as region of differentiation. Also known as region of differentiation, it has root hairs. Now region of elongation, elongation means the cells have elongated, the cells have enlarged, and they are characterized by a lot of vacuolation. So basically large vacuoles develop in the cells, that is why they get enlarged. Are these points clear? Then moving on to the region of meristem. Meristem cells are supposed to divide. So this region shows active cell division. Cells are small, thin, and they contribute to the primary growth. Are these points all clear to everyone? Please let me know that you are noting down these points. Please let me know that you are noting down these points. Quickly let me know. Yes, please. Are you all noting down these points? Just a second. Are you noting down all these points? Perfect. Now moving on to the root cap. See, the tip of the root is always covered by this thimble-like structure, and this thimble-like structure is known as root cap. Now by the way, root cap is also secreted by, meristem, meristematic cells. Now, what is the purpose of root cap? Basically, this root cap is going to protect our soft meristematic tissue, protects the meristem. So basically, now because of this root cap, the zone of meristem becomes subterminal. So basically, root meristem becomes not terminal, it is subterminal because of the presence of root cap. Are these points clear to everyone? Anyone with any doubt? Is that clear, guys? Don't worry about the notes. Don't get so much distracted. Please concentrate. Please concentrate, yes, on today's session. I have already uploaded the notes. Don't waste your time because I have told you multiple times it is very important that you make your notes yourself. Okay? Right. Then moving on to the root modification. See, I've got so many flowcharts for you so that things get easier to remember. So there are three types of root modifications. One for the purpose of support, second for the purpose of storage, and then we have for respiration. Is this clear? Is this clear till now? So see, for storage, we have prop roots number one and we have stilt roots. So prop roots we have seen are present in banyan. They are present in banyan, while stilt roots are found in sugarcane. They are found in sugarcane. They are also found in monstera. So where are these prop roots coming from? The prop roots are going to be formed from the lowest node. They develop from the lowest node of the plant. Is it clear? While the hanging roots, they develop from the upper branches, like in case of banyan. Upper branches. Are these points clear? Then coming on to the second type of root modification, which is tap root. So storage, for storage. So even the tap roots can store and even the adventitious roots can store. So the example of tap roots which are involved in storage, number one, carrot. Number two, radish. Are you getting that? Number three, we have turnip. And the adventitious roots which are involved in storage, number one, we have sweet potato. And we have asparagus. So you have to learn all these examples and also know which of these are tap roots storing the food and which ones are the adventitious. Now coming on to the respiratory roots. See, first of all, respiratory roots, also known as pneumatophores, they are only found in salty conditions, saline conditions, or marshy conditions where it is very difficult for the air to get trapped into the soil. So such roots have pneumatophores. Now these roots actually come out of the ground and they have these special holes on them, and these are no holes are known as pneumatothores. Holes are known as pneumatothores, while the root which comes out is known as the pneumatophore. So very important example of these. So which plants have these pneumatophores? It is rhizophora. Rhizophora. So these are the roots which are found in mangroves, in marshy areas. Are these points clear to everyone? Please let me know. Or there is a lot of water logging. So where there are mangroves or lot of water logging or the soil is very saline, right? Such have these pneumatophores. And now see, root is supposed to go towards the ground, right? But pneumatophores grow above the ground. So these pneumatophores, remember, these are negatively geotropic. These are negatively geotropic, while a typical root is supposed to be positively geotropic. Are these points clear to everyone? Please let me know. Are these points clear to everyone? Right. All right then. Moving on to next. Yes, see, this is something that I've already informed to you that 19th of April onwards, guys, from 2 to 5 p.m. are free, absolutely free. All India mock test series is beginning, and see, you have to practice these mock test series for real NEET exam experience. It's important that you are ready for the exam before going for the real exam. Also, you will get the instant all India rank prediction to get to know where you really stand. Also, you will be able to analyze your preparation. You will compete with lakhs of students, and then how to find out where did you make mistake. So there are video solutions available. So top 50 students will get mentorship after every student. Top 10 will get the WhatsApp guidance. So guidance is important, right? So where are you lagging? So teachers will be available for you. Just you have to come in the top 10 position. And then top 3 students are going to get Amazon voucher worth rupees 500. Alright guys, moving on to the next part. The next thing is STEM. So root is done. Next, coming on to the stem. See, unlike root, which is positively geotropic, stem is negatively geotropic. Stem is negatively geotropic. Are these points clear? Wait. There is some issue today. Anyways, just a second. Okay. So the stem is negatively geotropic while it is positively phototropic. It goes towards the light but away from the ground. Is that clear? So what are the characteristics of the stem? They are the ascending part. Ascending means it grows upward. It develops from which part? It develops from the plumule. It is green when it is young and it is brown and woody when it is old. Are these all points familiar to you? And are you able to recall? Now, what are the functions of the stem? Support. It bears structures like leaves and flowers and buds and all that, right? It conducts food and water. It is also involved in storage. For example, potato. What you eat is basically because it is storing food. Are you getting it? Right? And then it also gives support in the form of tendrils, right? In the form of tendrils, stem will be involved in giving support to the plant. And yes, it also leads to propagation. So any propagation without the use of seeds from roots, from stem and from leaves is known as vegetative propagation. So stem also acts as a vegetative propagule. Are these points clear? Now, what are the characteristics of the stem? What are the special structures that you get to see on the stem? Number one, stem always has nodes, right? Stem always has nodes. Now, nodes are those regions from where any other structure will develop. Leaves or tendril or thorn, everything or flower, everything is going to arise from this node, right? Now, between the two nodes, from 1 and 2, the space between the two nodes is known as internode, and nothing grows in here. Nothing grows in this region. Then we have axillary buds. So the axillary buds are the buds which arise from the axil. See, what is axil? Let's say this is the leaf, this space. So buds arising from this space are known as axillary buds, and the buds which are at the tip are known as the terminal buds. Are these points clear to all? Are these points clear to everyone? Please let me know. Now coming on to the modifications of stem. Quickly. Are you writing it along? Yes. So coming on to stem modification. See, the first stem modification we are going to talk about is tendril. What is the purpose of tendril? To give support. Now examples are super important. What are the examples of plants which have stem tendrils? See, please understand, we are also going to talk about the leaf tendrils. So you have to remember it clearly which examples are of stem tendrils and which are the leaf tendrils. I'll also give you a trick to remember. Now see, the stem tendrils, let's see. The stem tendrils are present in gourds. In gourds like cucumber. Please keep writing down these examples. Cucumber, then pumpkin, pumpkin, then watermelon. I don't know what's happening. It's not writing for me. Anyways, are these points clear to everyone? Cucumber, watermelon, and pumpkin. Pumpkin and watermelon. Right? Then another example is grapevine. Another example is grapevine. And then we have passiflora. Just a second, guys. Passiflora. So these are the examples of all the plants having stem tendrils. So you have to remember cucumber, pumpkin, watermelon, grapevine, and passiflora. All these are collectively known as gourds, right? Another stem modification is for the sake of protection. Now, the plants which have thorns, for example, bougainvillea. Remember, we also read about bougainvillea in homologous organs, bougainvillea. Now see, so interesting. All these stem modifications, all these structures, tendrils, thorns, all these cladode and phyloclad, all these they are homologous to each other. Why? Because they are all stem modifications. Isn't that so? So see, it is such an important understanding. They are all homologous organs to each other. Tendrils, stem tendrils and thorns and phyloclads, they are all homologous because they have the common origin, which is stem, but they will play different functions. And we can see the function played by tendril is support. Then thorns are meant for protection from browsing animals. Are these points clear? Then we have cladode and phyloclad. Are these points clear to here? Clear till here? Now, phyloclad and cladode, they are both meant for reducing the transpiration. They are both meant for reducing the transpiration. Now, what is the difference between cladode and phyloclad? See, phyloclad is a flattened leaf-like structure. Flattened leaf-like structure. Right? While cladode is a cylindrical and flattened green-like structure. Flattened green stem-like structure. Is this point clear? Now, cladodes always have limited growth. Cladodes will always have limited growth. Are these points clear to everyone? Yes, please. Are these points clear to everyone? Now, this particular slide is going to be super important for all of you. Citrus, yes. Thorn is also found in citrus. You're very right. So, thorns are also found in citrus. Right? Is this clear? Now, moving on to the next. This slide, guys, you can actually take a screenshot. Is going to be very important. See here. So these are also stem modifications. All these stem modifications have the function of vegetative propagation. Now they are all different from each other because of their origin and how they grow along, right? So first is runner. So you have to see the examples. In runner, we have strawberry and lawn grass. Stolen, we have jasmine and mint. This is not really given in NCERT, but just modification is written. So we don't know whether it is there or not, but I am definitely going to discuss it with you. It was given in old NCERT. Offset is water, hyacinth, and pistia. Then rhizome, the example is ginger, turmeric, and banana. And for sucker also, we have banana, chrysanthemum, and pineapple. Are these points clear? Now, what are the key features? What are the key features here? Yes, what are the key features here? So runner is a horizontally growing creeping stem on the soil surface with the long internode. See, you can see the diagram also. So this is one node, this is the second node, and see the internodal distance between the two nodes is very different, very large, and then nodes give rise to the new plants. So parent plant will often die, but the nodes will continue. The adventitious roots which grow on the nodes, they will continue to grow the plant. Are these points clear? Then moving on to stolon, lateral branch from the base of the stem will arise, which will grow upwards. So like this. See, this is the plant, right? This is the main parent plant. So this is the lateral branch which will grow from the base of the stem. It will bend downwards, touch the soil, and the part which touches the soil will start bearing roots. And this will form the new plant. So after a while, I can actually cut it from here and grow it as a separate plant. Coming on to offset, very short runner, one internode, and on every internode, there is tuft of roots and leaves. So see here, this parent, this is the picture of an offset. So at every node, we have tuft of leaves, group of leaves, and group of roots. Now, commonly seen in the aquatic plants and easily detachable. Example is the pistia and water hyacinth, both are aquatic plants. Coming on to rhizome, underground horizontally growing stem, just as in case of ginger and turmeric, right? And then we have sucker, chrysanthemums and pineapple, lateral branches which arise from the underground and grows obliquely. And they grow obliquely. Are these points clear? Are these points clear to all? Now coming on to leaf. Next part of this chapter is leaf. So we know that leaves are, listen, so we know that leaves are lateral structures. They are flattened structures. They are born at the node. They originate at the apical meristem, right? So if this is the stem, this apex is going to give rise to the new leaves. So leaves arise from the apical meristem. And now new leaves are always at the tip, at the apical meristem. New is towards the tip and the older one are towards the base. This is known as acropetal arrangement. New at the tip. Whenever anything grows, new thing grows at the tip, we call it acropetal arrangement. Is it clear? Now a typical leaf will always have three parts. A typical leaf will have three parts. See, this is a stem. This is the node from where it is going to grow, right? So what are the three parts? The three parts, this point of attachment is known as the leaf base. This is known as the leaf base. This stalk of the leaf is known as petiole, while this entire expanded portion is known as leaf blade, or we call it lamina. So lamina is another term for leaf blade. Are these points clear to all? Anyone with any doubt, please let me know. Yes, Ishornia is known as Bengal terror, not tiger. Are these points clear to everyone? Are you making notes with me? Please let me know. Are you writing all these notes with me? Everybody's getting it, right? Okay now. So now NCERT has described that leaf bases can be of three types. We can see three different types. We can see three different types at the or structures at the leaf base. So one is stipule, second is pulvinus, and third is leaf sheath. Now see, what is a stipule? Now see it with the help of a diagram. Let's say this is the stem, this is the node, and this is your leaf, right? But at the base of the stem, two leaf-like lateral structures, very thin leaf-like structures which arise at the base of stem. These are called stipules. These are known as stipules. While pulvinous, see here, pulvinous is a swollen base. Whenever the base is swollen base will be known as pulvinous. Okay, for example, stem, this is the base, this is the node, this is the leaf, but the base of the leaf is swollen. Whenever the base of the leaf is swollen, that is known as pulvinous. And coming on to leaf sheath. Now leaf sheath means this is particularly seen in monocots. Okay, now what happens in leaf sheath? So the base will surround the entire stem like this. So it forms a, if suppose this is the stem, so this base is going to form a round, it's going to form a wrap around the stem. So this wrapping around the stem is known as leaf sheath. Are these points clear to all? Are these points clear to everyone? Right? Okay. So leaf sheaths are always seen in monocots. Pulvinous. Yes, please. Anybody who can tell me where is pulvinous seen? Yes, guys. So stipules, we have pulvinous and we have leaf sheath. Are these points clear to everyone? Moving on to the next now. Next is leaf modifications. Now first is the leaf tendril. The function of leaf tendril will be support, of course. Now, the trick is to learn which plants have stem tendrils and which ones have leaf tendrils. So, for stem tendril, let's recall what we have written. Stem tendrils are found in watermelon. They're found in pumpkin. They're found in gourds, right? All the gourds, cucumber, pumpkin, all these are found in, all of these have stem tendrils. But for leaf tendrils, we must remember pea, beans. These have leaf tendrils. So how do I remember? Leaf is known as patta in Hindi, right? So P for patta, P for pea. And beans are also of the same family, so they come together, right? So this is how I remember. Patta has P. Leaf is patta and leaf tendrils are found in pea and beans. Are these points clear? Now tendrils are meant for, they are the reduced forms of leaf. Spines are the reduced form of leaves, right? And what are they meant for? For reducing transpiration. For reducing transpiration. So which all plants have spines? All the xerophytes. Not all actually. The xerophytes. For example, we can say Opuntia, right? And we can say cactus, Opuntia or cactus, right? One and the same thing. So xerophytes have spines to reduce the surface area and reduce the surface area to reduce the transpiration. So the leaves are reduced into the form of spines. Are these clear? Now talking about the leaves which can store food. So storage leaves. What do storage leaves do? They store food, right? They store food. Example, garlic. So basically the part of garlic that we eat or the onion leaves that we eat, they are leaves. Okay. Now insectivorous plants. In insectivorous plants, for example, pitcher plant or Venus flytrap, right? Venus flytrap. All these plants, flytrap, they have special structures to ingest the insect. Now, the part which gets modified for this purpose is actually a leaf. Okay, so the picture is formed from a leaf. So there are normal leaves which are photosynthetic in nature, and then there are these modified leaves which are going to ingest the plant. Are these points clear? Okay, then we have scale leaves. Now, in onion and garlic itself, see that dry leaf which you always peel off, the outermost leaf of an onion which you always peel off, which is dry, right? That is a scaly leaf. And then moving on to phyllodes. So phyllodes is basically petiole which gets flattened. So phyllodes is a modification of petiole which gets flattened and becomes photosynthetic. See, the function of photosynthesis is supposed to be performed by lamina because it is broad. It has large surface area to trap the light. But when petiole, which is the stalk of the leaf, that becomes flattened and becomes photosynthetic, that modification of leaf is known as phylloid. Are these points clear to all? Now the example of phylloid is acacia. The example of phylloid is acacia. Please let me know that all these points are super clear to everyone. Yes, please. Please let me know that all these points are clear to everyone. Okay, moving ahead then. Talking about venation. So what do we understand by venation, guys? What do we understand by venation? It is the arrangement of veins and veinlets. Arrangement of veins and veinlets. We all know this, right? So how is reticulate? Reticulate means there is a network which is formed. Parallel, as the name says, all the veins, they are going to run parallel to each other. So this is parallel, and in network we have a main vein which is known as midrib. This is known as midrib. The main vein is known as midrib, and then we have these smaller veins and then their veinlets. Is that clear? Anyone with any doubt till here? Quickly let me know. Quickly let me know, guys. So dicot venation is a characteristic of reticulate venation is a characteristic of dicots, and parallel venation is a characteristic feature of monocots. So they can also ask a question by giving the example of a monocot. So rice, wheat, bajra, sugarcane, bamboo, all these are monocots and they will have parallel venation, while the dicots will typically have reticulate. See, all dicots will have reticulate. Now that statement is wrong. Please understand. If somebody writes that all dicots have reticulate venation, or if we write that all monocots have parallel venation, now this is a wrong statement because there are always exceptions, just not in our syllabus. Are these points clear? Now coming on to types of leaves. So basically we have two types of leaves. One is simple, another is compound. See, how do you identify? How do you identify? So this is the node, right? If this is the node and this is the leaf, right? So every node is going to bear a leaf. Now see here. So this is the axil. This is the axil. So axil will bear buds. Axil will bear buds. Are these points clear to everyone? Now you will understand how to differentiate. So basically, simple leaf is the one whose lamina is not divided. I think I should not experiment a lot today with this. Lamina is not divided. Yes, lamina is not divided in case of simple leaf. Are these points clear? And in case the leaf is incised like this, then at least the incisions will not reach the midrib. Second point, incisions do not reach the midrib. Do not reach the midrib. Are these points clear to all? Yes, please. Are these points clear to everyone? Now coming on to compound leaves. So compound leaves can be of two types, pinnately and palmately. In pinnately, see what happens. Pinnately leaflets. The leaf is divided into leaflets. Leaf divided into leaflets along a common axis. A common axis known as rachis. So basically what happens, suppose this is supposed to be a leaf, right? But instead of one common lamina, now the lamina got divided into small leaflets like this. So the whole lamina has been divided into smaller leaves known as leaflets. We don't have a concept of leaf here. These are leaflets. The total thing is known as a leaf. These are the leaflets. Now, how do we differentiate? I told you at the base of every leaf is a bud. So, the bud would be present here. This is where the bud should be present. Are you getting it? So, the entire region beyond this is a leaf, while the smaller ones are known as the leaflets. Please let me know that this point is clear to all. So most important point is to remember the example. The example is neem. Even rose has pinnately compound. Now coming on to palmately, the example will be silk cotton. Now what happens here in silk cotton, basically, so the incisions, see, this is the stem, this is the node, this is the petiole, this is the tip of the petiole, and all the leaflets are arranged at the tip of the petiole like this. This is palmately compound. Please let me know that all these points are clear to you. Yes, please. Quickly let me know that all these points are clear to everyone. So the characteristic point of palmately is that leaflets are arranged at the tip of the petiole. See, that day also, in while we were studying our unit 1, I told you there are certain characteristic terms. The moment you hear that, quickly that thing should come to your mind, right? For example, when we think of pseudocoelom, we think of esculmenthes, right? When we think of diatom, soapbox word comes in, right? Similarly, if somebody says leaflets at the tip of the petiole, you have to quickly mark down the answer. It is palmately compound. And what is the example? What is the example? Silk cotton. Ma'am, no stem and root modification. Who is this? Who is this? No root and modification, stem modification. We have already done that. Already discussed. Yes, please. Now coming on to phyllotaxy. See, what is phyllotaxy? Yes, please. What is phyllotaxy? Yes, please. All right. So talking about phyllotaxy, phyllotaxy is the arrangement. It is the arrangement of leaves on stem. It is the arrangement of leaves on stem. So how are leaves arranged? For example, in alternate, guys, in alternate, see what happens. See, let's say these are the nodes. So if this has one leaf, another leaf will be present on the alternate side like this. And so one node has one leaf, one node, one leaf. This is the underlining statement of alternate phyllotaxy. Are these points clear? While in opposite what happens? Every node will have two leaves on the opposite side. The two leaves will be opposite to each other. So one node, two leaves. Are these points clear? And then talking about word, in word basically, one node is going to have a circle of leaves, a whorl of leaves. So one node, many leaves. Are these points clear to everyone? Please let me know. There is no doubt. Quick, quick, quick. Now talking about the example. So for opposite, see, we have example kala gura. Kala gura, kala means black, gura means white. So they are opposite to each other. So how do we remember? Kala is calotropis and gura for guava. So calotropis and guava, these are the two examples. For word, we have Elstonia. For word, we have Elstonia. And for alternate, we have China, rose, mustard, and sunflower. China rose, mustard, and sunflower. These are the examples of these phyllotaxy. Please let me know that these points are clear to all of you. Yes, please. Are these points clear to all of you? Yes, yes, yes. So those of you who remember the examples, fabulous. And you have to check all this memory in your mock test now. See, it's very easy to sit relaxed and, attempt the questions. Everything falls in place. Everything comes back very easily. But when you are panic stricken, when you are in that, exam surrounding, then things become different. So in order to prepare yourself, start with your mock tests from 19th onwards. Okay. Now coming on to inflorescence. See, inflorescence is the arrangement of flowers on the floral axis. How are flowers arranged on the floral axis? Okay. So that is known as inflorescence. Uh-oh. And remember that flower is a modified shoot. So basically what do we say? That Thalamus is a region where all the nodes, they will condense and internodes become very small, and different parts are going to be produced on this thalamus. So this is your pistil. Here we have these stamens. These are also modified. These are all modified leaves. So flower is called the modified shoot, and all the floral appendages are known as the leaf modifications. They are all the leaves have become the floral appendages in case of flower. Is this clear? And now this is a petal, and then we have these sepals, right? So basically all these floral appendages are condensed together on thalamus. Are these points clear to everyone? Modification of leaves done. Pop, what's happening? You're lagging behind. We can't afford lagging behind now. All right, guys. Now, the pattern in which the inflorescence can be seen, there are two patterns in which the inflorescence appears. One is the racemose inflorescence, and second is the cymose inflorescence. So, what do we understand by racemose? See, I always remember it. Racemose means the tip is always going to race upwards. So, tip continues to grow. Tip continues to grow. Is this clear? This is race mode. So main axis will continue to grow. So basically the flowers are in acropetal manner. Flowers in acropetal succession. What is acropetal? What is acropetal? New at the tip. So when new is at the tip, that is called acropetal. Is that clear? And this is the racemose type of inflorescence. While in cymose, tip stops growing, tip stops growing, and flowers are arranged. Flowers. Wait a second, guys. Yes, flowers are arranged in the basipetal manner. So it's easy to remember. Basipetal means new towards the base. Is this point clear? The new or the younger ones, the young or the new at the base. Easy to remember. Easy to remember. Yes or no? Quickly give me a thumbs up that it's all easy to remember. Quickly let me know. Yes, please. Easy to remember? You have to tell me in the comment section later on, guys, that things were easy for you to remember this way. Yes, please. Is that clear? Clear to all? Now moving on to the next part, which is flower in detail. See, flower is known as the reproductive shoot or the reproductive part of the flower. And we have just read that it is a modified shoot where the leaves have been modified into the floral appendages. So leaves have modified into the floral appendages. And there are four types of appendages. These are four in type. So four whorls. What are these? Sepals. The collective term for sepals is calyx, right? Then we have petals. For petals, we call it corolla. For stamens, the male reproductive part, which is known as androecium. Andro means men. Yes or no? And then we have pistil, which is called gynoecium. Gyne word is for female. Please let me know that all these points are clear. What is the difference between calyx and sepal? Calyx is a collective term. All these are the collective terms. Please let me know that these points are super clear to all. Yes, please. Clear? Alright, moving on to now floral symmetry. Very, very important. Very, 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 very important. Every year we get a question from this topic. Every year. Either they are going to ask you about actinomorphic, the examples of actinomorphic or zygomorphic. First, let's understand. See, actinomorphic means a radial symmetry. Means you can divide the flower in equal halves along any plane. Okay. And what is zygomorphic? Just like in animals, we talk about bilateral. So in flowers, we use the term zygomorphic. So we can divide the flower into equal halves along only one plane. In radial, along any plane, and in bilateral, along only one plane. Is it clear? And if the flower cannot be divided into into two halves along any plane, we call it asymmetry. So no symmetry at all, cannot be divided into two equal halves. Now the examples for asymmetry is kana. The example for asymmetry is kana. Is that clear? Although it is rare, but it is seen in kana. Now talking about actinomorphic. So the examples are datura, mustard, and chili. So how do I remember? Datura must chill. Datura must act chill. See, datura must act chill. So how do I remember? Datura must say mustard, chill, say chili. Act word is for, it tells you that it is the example of actinomorphic. See, sometimes, do you know what happens? We, of course, it is biology. There are so many examples and we make so many mnemonics that sometimes we forget that which mnemonic was for which yes, so we forgot that which mnemonic have we made for which purpose. So it is important that number one, you don't make mnemonics with the first letters because see, C can stand for chili or cotton or China rose or cassia. So always make full word mnemonics, right? The tura must chill. It must act chill, right? And then zygomorphic. For zygomorphic, we have cassia, cassia, ne, gulmohar, pk, P, K, bean, vagina. So this is how I remember. So cassia is cassia, gulmohar, then P and B. Is that clear? So cassia, gulmohar, P, bean. These are the examples of zygomorphic. All clear to everyone? Please let me know. See, there are many mnemonics coming up. I know this chapter is full of examples. It is difficult for you. So I'm trying to make it easy for all of you. Yes, please. All clear? Everyone? Good evening. Those of you who are late, you will have to attend the session later also. But right now, just go on with this. Go on with the flow. Are these points clear to everyone? All right, guys. Now, on the basis of number of whorls, we have three types of flowers. Trimerous means everything is in the multi 3 or in the multiple of 3. 4 or the multiples of 4, tetramerous. And then 5 or 5, the multiples of 5, we will call it pentamerous. Is that clear? Now, moving on. Type of flowers. Very, 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 very important. How many times should I write this? Every year you get question from this topic. Every year. Now see, first we have on the basis of, what is the class basis of classification? Position of ovary. See, last unit also I told you the basis of classifications are very important. So is here. So types of flowers on the basis of position of ovary. We have three different types of flowers. One is hypogynous, we have perigynous, and we have epigynous. So what is hypogynous? Let's split the word. Let's split the word. In case you forget, how do you reduce it? Hypo means below. Everything is below the ovary. So basically, ovary has the highest position. Everything below ovary means ovary is at the top. Ovary is at the topmost position. So such ovary is known as superior. Such ovary is known as superior. Are these points clear to all? Are these points clear to everyone? So superior ovary. Is this clear? So ovary is superior here. Talking about perigynous. Peri means around the ovary. So basically, ovary is not superior. Ovary is known as half inferior. Everything is at the same level. Okay, half inferior. And then epigynous, above the ovary. So basically, if everything is above the ovary, so ovary is at the lowest position. Yes or no? Ovary, the lowest position. Yes, please. Ovary holds the lowest position. So we call such ovary as inferior. So it's very important that you remember this. Superior ovary. Wait, what is happening? Today the tab is getting slightly mischievous, getting bashed later on. Now, this is the superior ovary. So, perigynous always has the half inferior ovary, while epigynous will have inferior ovary. Are these points clear? Are these points clear to all? So, hypogynous will have superior ovary, perigynous will have half inferior, and epigynous will have inferior ovary. Are these points clear to all? Now, time to remember the examples. See, how do we remember the examples? So, my trick is he must bring China rose. He must bring China rose. So what is the example? Or he must bring superior China rose. So everything gets remembered here. So he for hypogynous, M for, this is example, mustard, bring for brinjal, and China rose. So he must bring superior China rose. So you remember that what are the examples, and also you remember the hypogynous always has superior ovary. Are these points clear to everyone? Please let me know. Mnemonics will be in English only. He must bring superior China rose. Mostly it is in English. Then perigynous. So how do we remember the examples for perigynous? People pluck. People pluck peach and rose. Is this clear? People pluck peach and rose. So the example is plum, peach and rose. Are these points clear to all? Please let me know. Yes, please. Quickly let me know that all these points are clear to everyone. Yes, please. All points clear? Okay, guys. Moving on to the next then. Moving on. So people pluck peach and rose. And for epigynous, we have, yes, please. For epigynous, epic, sunny, cooks guava. Epic, sunny, cooks guava. So how do we remember? For epic, we have epigynous, sunny is for sunflower, cooks is for cucumber, and guava is guava. All these examples learned. Also, you have to remember that ray florets of sunflower will show this kind of flower. Are these points clear to everyone? Please let me know. Please let me know. Yes, please. GCS, Arnita, yes. See, first of all, there is no standard for making any mnemonic. But my personal experience is when you make such GCS kind of mnemonics, right? So what happens during your exam? It won't happen now. Trust me on this. It won't happen now. But in your exam, suddenly your third eye will open, and suddenly you will get to think. You will think, oh my God, C could be China rose. C could be cassia. C could be chili also, because you have all these examples in your syllabus, right? You have all these examples. So my personal advice to all of you is that make mnemonics which have full word or at least half the word to give you an idea the direction that what does that alphabet stand for. I personally feel this. You accept that it happens. See, I've been in your shoes once. So suddenly, the things that you have never thought of, the kind of research oriented brain that we have never had in these 2 years of preparation, suddenly, I don't know how it will awaken. And all we are going to think about is, okay, this could also happen. So I feel let's keep it some mistake proof, and go ahead with the mnemonics. Now coming on to estivation. See, estivation, valvate estivation. So most of the questions from this topic are example oriented, right? They are all example oriented. Let's talk about estivation here. Now, in valvate estivation, the example is calotropis. It is calotropis. In twisted, we have cotton, China rose, and ladyfinger. And ladyfinger. For imbricate, guys, we have gulmohar and cassia. And for vexillary, the example is beans and pea. Is that clear? So in valvate, what happens? So they are joined margin to margin. There is no overlapping at all. So the key word is no overlapping. No overlapping at all. So margins just meet, they don't overlap. No overlapping. This is the example. In twisted, so one corner is inside, other is outside. One is inside, other is outside. Like this. Are you getting that? So this is twisted. It can be 3, it could be 4, it can be 5 also. Right? So twisted. So what is the key point here? Overlaps. Overlapping. Overlapping occurs in a fixed pattern. Okay, overlapping occurs in a fixed pattern. While in imbricate, what happens? Overlapping is there, but no fixed pattern of overlapping. No fixed pattern of overlapping. Are these points clear? And then talking about vexillary. See, vexillary deserves a separate page. So in vexillary, what happens? We have 5 petals, first of all. We have 5 petals like this. This is the largest one. We call it standard. This is vexillary I'm talking about. Vexillary can also be known as papilionaceous. Papilionaceous. Right? Also known as papilionaceous. So we always have 5 petals in this. So the largest one will be known as standard. Then the other 2, these are on the sides, so we'll call them laterals, like this. And the smallest ones on the inner side, like this. These are known as keel. Now the 2 keels, they are fused. They are always fused. So we have 1 standard, we have 2 laterals, and we have 2 keels. Now they are fused, so we'll always put a bracket. So whenever we have to write down the formula for vexillary estivation, it is always 1 plus 2 plus 2 in brackets. Is this clear? Also remember that this is the anterior side and this is the posterior side. So standard is towards the posterior. So this question can appear. They might give you a statement based question, right? That the largest petal, which is known as standard, is towards the anterior side. The statement goes wrong. Is that point clear? Now, can you solve such questions? Can you solve such questions? Yes, please. Moving on to the next. Now, talking about the conditions. See, what are the different conditions that can appear for calyx, for corolla? Let's quickly talk about them. See, if the calyx is fused, we'll say gemosepalous. Calyx is sepals, right? So for fused calyx, we say gemosepalous. So gemo word is for fused, right? And if they're free, we'll call them, yes please, if they're free, we use the term, what term do we use for the free stamen? Sorry, free sepal, quickly let me know. Poly, yes, very right. Polysepalous. Poly is for free. Similarly, corolla is for petals. So if they are fused, we'll say gamopetalous and polypetalous. Then for androecium, we have few different terms also. See here. For androecium, androecium is a collective term used for stamens, right? Now see here, what are the different terms we are going to use? If the stamen is sterile, if it is sterile, we'll call it staminode. If it is sterile, we'll call it staminode, right? If it is free, if they are attached to the petal, we are going to use the term epipetalous. We are going to use the term epipetalous. If it is going to be attached to the perianth, then we'll call it epiphyllous. This topic is not just about the terms. We'll be inserting the examples here. Then androecium. In case androecium, so there are, let's say, 10 androecium, right? Now all the 10 androecium, they are united. So first of all, if they are free, we are going to use the term, yes please, polyandrous. If they are all free, polyandrous. And if they are fused, we are going to use the term synandrous. Syn means fusion. Now these synandrous can be of further types. It can be monoadelphous. It can be diadelphous, means all the 10 will group together to form one unit. So 10 with brackets. Diadelphous means they are going, di means 2, they are going to form two groups. Now it could be 5 plus 5, it could be 9 plus 1, like this. Are you getting that? So this is diadelphous, or they can form three groups, and more than two, we'll just call it polyadelphous. Is that clear? So it could be 2 plus 3 plus 5. So this is polyadelphous condition. Are these points clear to everyone? Now we have to remember the examples in which each condition is found. See here. For monoadelphous, just remember we have Malvaceae. For monoadelphous, M for monoadelphous, M for Malvaceae. Now what are the examples of Malvaceae? Cotton, China rose, bhindi, okra, right? For diadelphous, we have P. Diadelphous is found in Fabaceae. Pea, beans. Are you getting that? And polyadelphous is seen in citrus. Clear? Similarly, we have examples here also. Yes, please. For epiphyllous, we have two examples. Lily. We have example of lily. This is epiphyllous. Please let me know that all these points are clear to you all. Quick, quick, quick. Please let me know that all these examples are clear to you all. Right. Okay. Yes, please. So are we able to recollect and recall all the examples that we have learned in the entire 2 years? Yes, please. All right, moving on to gynoecium. Similarly, gynoecium can be, if there are more than one pistil, then we can call it, yes, please. Everything clear? Anyone with any doubt? Anyone with any doubt till now? See, all these terms, polypetalous, polyadelphous, these were in the year 2024. This was in 2024. This one, these all terms, these were monoadelphous, then syncarpus, I'll come on to that, epipetalous, polyadelphous, this was in 2024. Staminode was in 2024. Clear? Now, coming on to, so we have, if more than one carpel is present and they are free, we can call it epocarpus. Epocarpus for free carpels. And if they are all fused, we'll call them syncarpus. Clear? If they are fused, we'll call them syncarpus. Are these points clear? And if there is one ovary, we'll call it monocarpellary, right? If there is two for one ovary, or sorry, one carpel or pistil, we'll call it monocarpellary. For two, bicarpellary, and so on. Is this clear? One will always be epocarpus. Two can be syn as well, or epo also. So depends. Please let me know that all these points are clear to all of you. Yes, please. All these points are clear to you. Now moving on to the placentation. Very important aspect. Very important. See, we have marginal. For marginal, the example is P. For exile, we have lemon and we have China rose. For parietal, we have two examples again. We have argemone and we have mustard. For free central, we have again two examples, primrose and dianthus. And for basil, again, marigold and sunflower. Clear? Now, what are the key indications? How do you detect that which kind of placentation it is? What is placentation? The arrangement of placenta. So the key words that you have to remember. In marginal, guys, there is always simple and unilocular ovary. It is always simple and unilocular, and they are arranged at the margin. They are at the margin. Ovules arranged, marginal ovules, where there is ventral suture. So these are the key words to remember. Moving on to exile. In exile, there is always a central column around which they are arranged, and it is always a multilocular ovary. It is always a multilocular ovary. These are the key words that you have to remember. For parietal, they are all around the wall. So ovules at the inner wall of ovary. Inner wall of ovary. Then for free central, they are at for free central, septa absent. See, in case of exile, multilocular ovary, septa is present, true septa present. In parietal, you also have to remember there is a false septum. And in free central, septum is absent. It is unilocular. It is a unilocular ovary, and all the ovules in the center. And for basal, at the base. Please let me know that all these points are clear to you all. All these points are clear to you all. Please let me know. Yes, please. Please let me know all these points are clear to you all. Tomato also comes under, yes, tomato absolutely comes under this category, exile, tomato. Is that clear? Okay. Moving on to the next point, that is fruit. So we have three types of fruits. First of all, fruit is a ripened ovary. We all know that. But may not be. Depends which type of fruit are we talking about. If we say that fruit has developed from the ovary, it is called the true fruit. So it develops from ovary. Develops from ovary. If it develops from any part other than ovary, it will be known as false fruit. And parthenocarpic does not develop as a result of fertilization. So no fertilization, so no seeds. So parthenocarpic fruits are always seedless. Okay. Now we have to remember the example also. So any part other than ovary, usually it is thalamus. Yes, usually it is thalamus. Now let's talk about the true fruit first. Guava, mango, these are all true fruits. From part other than ovary, usually thalamus, example is apple, cashew, strawberry. These are all examples of false fruits. And for parthenocarpic, we have an example, banana or grapes. We can develop seedless varieties also by this. Are these points clear? Arnita, how to remember the examples of are fine, but this ovule. Which ovule are you talking about? See, you can't make mnemonics for everything, right? Okay. Now coming on to the last part of this chapter, which is seed. So seed develops from ovule. It develops from ripened ovule. So ovary will become fruit and ovule will become seed, right? So seed is going to have two parts. One is the seed coat and second is the embryo. So there are two seed coats. Outer one will be known as testa, which is hard. The inner one is known as tegmen, which is the softer one, right? Testa and tegmen. Then embryo consists of three parts: radicle, which will give rise to the root system. Okay, and later on plumule will develop. The first embryo consists of radicle, which will give rise to the root system, embryonal axis, okay, from where the embryo will develop actually. And then cotyledons, they can be one or two in number, one in case of monocots and two in case of dicots. Are these points clear to everyone? Tomato. See, Hayota is asking, what about tomato? See, tomato has seeds. So normally tomato is not a parthenocarpic fruit, but we have read it in plant growth hormones chapter that we can induce parthenocarpy in tomato by the use of auxins. That can be induced, but naturally the tomato has lot of seeds, right? Now guys, coming on to floral diagram. What is a floral diagram? Instead of describing everything, we'll just show it in the form of symbols. So you have to remember these symbols. For bracteate, sepals, we use K, petals C, perianth. Perianth is when the petals and sepals are indistinct, right? And one unit of perianth is known as tepal. So androecium A, gynoecium G. Now, if gynoecium is superior, so in that case, we use a bar below it. This is for superior. So you have to remember the opposite way. A bar below means superior ovary. A bar above it means inferior ovary. Inferior or half inferior, both are represented by the same symbol. Is this clear? So you have to remember in the opposite way. Superior means up, right? But here the bar will be below. So then male plant, the symbol will be this. Female plant, this is the symbol for bisexual. We'll combine the two. For actinomorphic symmetry, we use this plus sign in the circle. For zygomorphic, we use the sign of percentage. And for fusion, how do we use it? We use the brackets, right? So you put the number of a particular unit and put bracket around it. This means fusion. Is this clear? Now, talking about the floral diagram. So, floral diagram means arrange. It illustrates the number as well as the arrangement of flower parts, their relationship, and the position of mother axis also. What do we understand? So, when you see a floral diagram, right? This is the floral formula. These are the symbols for floral formula, sorry. So for floral formula, we use a symbolic representation. Now in case of floral diagram, you will see this dot. This dot represents the mother axis, right? This represents the mother axis. That means towards which the stem is present. Is that clear? Right. And then, see, let's practice one. How do we write a floral formula once? So first of all, we write whether a bract is present or not. It could be bract or it could be e-bracteate. It could be bracteate. If the bract is present, bracteate. If it is not, then it is e-bracteate. Then we write the sexuality. So whether it is male plant or whether it is female plant, or we can write that it is bisexual. So we put this sign. Then we write the symmetry. It could be actinomorphic or it can be zygomorphic, right? And then we start writing the sepals, then petals, then androecium, and then gynoecium. Is that clear? Now you might have seen a cap like this. This cap shows the fusion of one whorl with another. So what does this cap represent? So fusion of androecium stamens with petals. Are these points clear? Right? So whatever the number is, you keep on writing. Let's say there are 5 petals and they're fused, write it like that. If the petals are 5 in number but they are not fused, you don't put a bracket. Then let's say there are 10 androecium and they are all free. You write 10. Let's say if there are 10 and they're all fused to form one unit, that is monoadelphous condition. This represents the monoadelphous condition. So you might get questions where you're given a formula and you'll have to answer what kind of condition can you conclude from that formula. Then also G, if the bar is below, it means it is a superior ovary, isn't it? Then how many ovaries are present? Like this. 2 bicarpellary condition and fused. So syncarpous and bicarpellary. Will you be able to answer such questions? Please let me know. Will you be able to answer such questions? Please let me know. Yes, fusion of androecium with petals. Now this condition is known as epipetalous condition. Absolutely. Now, in case petals and sepals are not distinct, we call it perianth. Then we use the term P for this. Is this clear? How many perianth? 5 and 5 fused. And let's say there are 10 androecium and these 10 androecium are fused with the perianth. What condition is this? Epiphyllous. Is that clear? Then let's say it has a inferior ovary, so bar above. And let's say there are two carpels and they are not fused together. So this is bicarpellary and apocarpus. Please let me know that you are thorough with this concept. Can we do that now? Can we do that now? Please let me know. Are these points clear to all? Perfect. And last not the least, guys, we are going to talk about dicotyledonous seed and monocotyledonous seed. What is the difference? So, in dicot, the seed is made up of 2 layers, outer testa, inner tegment, or while in monocot, the seed coats will be fused with the fruit wall, will be membranous. Do you remember the structure of maize seed? Now, the outer structure, this membranous sheath which is present around the seed, this is not just the seed coat, it is the fruit wall also. This is seed coat and fruit coat fused. Fruit wall fused together. What is the fruit wall known as? Pericarp. Yes or no? It is known as pericarp. Now there is the scar known as hilum, while there is no hilum here. So micropyle, the opening of the ovule, is a pore above the hilum. Is that clear? While here, micropyle is not visible. So in dicot, there are 2 cotyledons, while there is 1 cotyledon, and that 1 cotyledon is known as scutellum. So cotyledons store food. So cotyledons are basically seed leaves, which will store food. Right? Why do we need 2 seed leaves, 2 cotyledons here? Because there is no endosperm. This is a certain reason. There are 2 seed leaves here because there is no endosperm, and that is why we call them non-endospermic seeds. Mostly dicot seeds are non-endospermic. Only non-endospermic? No. Castor is an endospermic seed. Castor is a dicot, but it has endospermic seed. Is that clear? Similarly, here in monocots, endosperm is bulky. It is there. It stores food, right? So we don't need 2 cotyledons. We just have 1 cotyledon. And also you know that there is eleuron layer. Remember? So this is all endosperm. Yes or no? This is all endosperm. And then this layer is known as, this whole layer is the eleuron layer. Remember? This is the eleuron layer. Clear to all? And they are mostly endospermic. Orchid is the example of a monocot which is non-endospermic. Then in dicots, plumule and radicle are present at the 2 ends, while here they are tilted, number 1. And plumule is enclosed by a sheath called colluptile. Radicle is covered by a sheath known as collurizа. So collurizа and colluptile are absent here. These 2 structures, they are absent here. They are only present in the monocots. Please let me know that these points are clear to all of you. Yes, please. All points clear? And guys, once again, let me remind you that on 19th April, we are starting with a free mock test series, which you can't afford to miss. 2 p.m. to 5 p.m. for the real NEET exam experience, so that you don't panic in the final on the final day. So it is very, very important that you appear in all these mock tests. You will get to see your all India rank also. You'll compete with lakhs of students. You'll get your video solutions also to improve. You'll get 50 top 50 students will get mentorship also. So what is better than talking to the teachers directly? It actually calms your nerves, right? Then top 10 will also get the WhatsApp guidance, and top 3 will have Amazon vouchers. But the most important thing, you will get that real NEET exam experience. That is the most important. And with this, we start the next chapter, that is animal tissue. We are going to start with the next chapter. Please give me a thumbs up. Families, yes. Talking about families, the 5 families I will be revising in a quick session which will be of 10 minutes or 15 minutes, and I'll be revising all the 5 families in a separate video. You will get it by tomorrow. So guys, cockroach, frog and families, they will be all taken care of in that 10-minute video, and that is how we are going to revise. Okay, all right guys, moving on to the next chapter, that is animal tissues. So we know there are 4 types of animal tissues: epithelial, muscular, nervous and connective. Talking about epithelial tissue, what are the characteristics of epithelial tissue? It covers the surfaces. It is present on the body surface, internal as well as external. So it forms a coverings. Then they are always tightly packed. They are always compactly packed. You can say tightly or compactly packed. NCERT uses the word compactly. Then, in case there are intercellular places, then they are very minimal, very little intercellular spaces are present. Now, we know that there are 2 types of epithelium, right? So, epithelium, if it is in one layer, we call it simple epithelium. If it is multi-layered, then we call it compound epithelium. Are these points clear, everyone? Everyone? So simple epithelium will line the body cavity. Okay, please listen. Simple epithelium will line the body cavity, the ducts and the tubes, while compound epithelium will be actually protective in nature. Simple epithelium can also be involved in diffusion. Diffusion can only occur through simple epithelium, and that too only through squamous type, right? Simple epithelium will not really participate in protection. No protection. For protection, it has to be multi-layered. Are these points clear? So that is why compound epithelium. Points clear to everyone? Guys, quickly share the session also with your friends. Don't forget to do that. Like, subscribe and share. That is the drill. So everybody has to do that. And in case you are liking it, please let me know in the comment section as well as in the chat. In case there is a doubt after the session, please leave your doubt in the comment section. I will read the comments and answer to your question. And everybody has to join the Telegram channel for all the notes. The previous one have been updated today only. Okay, they have been updated today, and these will be immediately pinned in the Telegram channel. Are these points clear? Shall we move on? So moving on to now the simple type, the different types of simple epithelium. Simple epithelium can be of squamous type, cuboidal type, or columnar type. There is one more pseudostratified. We'll talk about it. So, see, thin layer of flattened cells. What is squamous here? Squamous means, wait. So, squamous are like, wait. Again. Okay. So, what are squamous like? Every time I try to change the color, Pratik, it's not time for deep explanation, that it's time for revision. If you want to attend deep explanations, you can watch other lectures already there uploaded here on the session on the channel. But these are quick final revisions where we are hitting on every word. We are recollecting every word. Okay. Now squamous epithelium. These are the flat cells like these. Cuboidal are cube-like cells like these with a central nucleus, and columnar are long and slender cells, brick-like cells. Are these points clear to all? Now reading about squamous, see, they are thin layers of flattened cells. They form irregular boundaries. And where are they present? They are present in the walls of blood vessels and alveoli. So they are so thin that they can actually help in diffusion. So they form the diffusion boundaries. Are these points clear to all? They form the diffusion boundaries. Then in cuboidal, the cells are cube-like and there is a central nucleus as said. And where are they present? In the ducts of glands, tubular parts of nephron. Okay, in PCT, DCT, all of that, in loop, yes, they have in loop of Henle, they also have cuboidal. So what is the function played by them? It is secretion and absorption. Now, in PCT, to enhance the role of reabsorption, there are microvilli present. So what do we understand by microvilli? So this free surface is modified into brush bordered like appearance. This is brush border. So whenever there is a brush border, it is always to increase the surface area for absorption. So where is brush bordered cuboidal epithelium present? That is PCT. Then moving on to columnar. Columnar is found in your stomach and intestine, again for secretion absorption. And now see, be it cuboidal or it is columnar, both these cells can be ciliated in nature. Whenever it is ciliated, cilia always helps in movement of something. Cilia always helps in movement of something. So ciliated cuboidal, see, ciliated and cuboidal, they are present in bronchioles, that is your respiratory tract, but wait a second. But ciliated and columnar, they are found in your fallopian tubes. So function of cilia is always to put something in motion, helps in movement. Are these points clear? All right, all right. Moving on to next. Now we have this, wait a second. We have one more type of simple epithelium, which is pseudostratified. There's some glitch in the, this is a very heavy PDF basically, because there are 3 chapters and I've made notes for 3 chapters all in one. Wait a second. Yes, please. Now coming on to pseudo stratified. Please let me know that there are no doubts. Pseudo stratified. Yes, please. Pseudo stratified means the cells are of different lengths, but in single layer. And you see the pseudo stratified in the respiratory tract. So there are some long cells, there are some short cells, but they are in one layer, but they appear to be multi-layered. Is it clear? They appear to be multi-layered. Are these points clear? Now coming on to the second type, and the second type is known as glandular. So first is simple, second is glandular, and the third one is compound. Wait. So where we have written, just a second guys, it is lagging a little bit more. Okay, I'll tell you here only. So we have simple epithelium, we have compound epithelium if it is multi-layered, and we have third secretory type of epithelium, and then that is known as glandular. Are these points clear? Now, glandular epithelium can be divided into 2 types on the basis of number of cells. On the basis of number of cells, there are 2 types. If it has one cell, it is made up of one cell, we'll call it unicellular epithelium or unicellular gland. And if it is made up of multiple cells, multiple cells, we'll call it multicellular gland. Now the examples are important. Unicellular gland is the goblet cell, and for multicellular we have salivary glands. Points clear? But we can also divide it on the basis of duct. So if we divide glandular epithelium on the basis of duct, we have 3 types: exocrine, duct present. Endocrine, no duct. So that is why we call them ductless. Heterocrine, they are both exocrine plus endocrine in nature. Are these points clear to all? Are these points clear to everyone? So example of exocrine, your salivary glands, your tear glands, sweat glands, mammary glands, they are all exocrine. They all have ducts which will carry the secretion. While we have read the whole chapter on endocrine, they are ductless glands. They release hormones. So all those hormone secreting glands, thyroid, parathyroid, they are all endocrine in nature. While one gland that you read about in that same chapter is pancreas. Now, pancreas is heterocrine because a part of pancreas is exocrine. Basically, 99% part of pancreas is exocrine and secretes juices, while 1% part of pancreas is endocrine. And that endocrine part is known as, quickly let me know in the chat. What is the endocrine part of pancreas known as? Yes, please. What is the endocrine part of pancreas known as? Aisles of Langerhans. Remember? Aisles of Langerhans, alpha, beta, delta cells. Right? Yes. Now coming on to cell junctions. See, cell junctions are mostly in epithelial. I did not say they are not present in other, but they are mostly seen in the epithelial tissue. Is that clear? We have three types of cell junctions to talk about: tight junctions, adhering junctions, and gap junctions. See, as the name says, tight. When you tighten the bottle, see how to remember. When you tighten the bottle cap, it prevents leakage. So similarly, tight junctions, they will prevent leakage. Adhering word means comes from adhesive, means cementing. So it joins the two cells together. And gap junctions, they facilitate communication. So wherever there is a gap, there comes a gap junction to help the two cells from far from each other to communicate with each other. So what is the communication like? It helps in the transfer of ions and molecules. Are these points clear to everyone? Are these points clear to everyone? Right. So these are the three types of cell junctions which will be found in all tissues but majorly in the epithelium. So guys, these are the NCERT pictures. What is this? This is a squamous epithelium. This is the cuboidal epithelium. This is the columnar epithelium. And this is the cilia. So this is the ciliated epithelium. And while this one is the third type of epithelium, multi-layered, we call it compound epithelium. Right. Now, talking about compound means they will be found in multiple layers, so they can be protective in nature. Your skin represents that. For example, skin. Is this clear? Now, compound epithelium can be basically of two types. It can be stratified if it is multi-layered. It can be stratified, then it will be protective. And second, we call it transitional epithelium. Now, transitional epithelium allows stretching. It allows stretching of the organs. So if you see transitional epithelium around your urinary system, in urinary bladder, it has to stretch. Urinary bladder, then your ureters, they have to stretch. So since this transitional epithelium is found around the urinary structure, we also call it urothelium. So they are so designed that they allow stretching. Now, one common feature of all these epithelial tissues, different types of epithelial tissue that we have talked about, is they all have this one layer which is known as, yes, who can tell me what is the common feature in all these epithelium? What is that unique feature? What type of membrane is present? Yes, please. Anyone who would like to tell me in the chat. So, what is the unique feature? Quick, quick, quick. Which is present in only epithelium, but in all the epithelium. What is the name of that non-living membrane that I'm talking about? Yes, please. Quickly let me know. Until that time, I hope my tab will also start working. Yes, please. Basal lamina. Do you remember? What do we call that? Basement membrane. Perfect. They all have one common membrane known as basement membrane. Yes, please. Uh-oh. Oh God. Please start working. Basement membrane, absolutely. So basement membrane is a characteristic feature only of epithelial tissue. And now this basement membrane is a non-living layer. And it is secreted by both epithelial tissue and connective tissue. Is this point clear to everyone? Is this point clear to everyone? All right. Clear to all? So moving on to next, wait. Moving on to next tissue, which is types of muscles. Now see, wait. See, there are three types of muscles. One is the voluntary muscle. The first diagram that you see in here is the voluntary muscle, right? The second diagram that you see is the smooth muscle or the involuntary muscle. And the third diagram represents the cardiac muscles. So first of all, you must know that where are these muscles present and what are the unique features of each type of muscle. So this is the this is the skeletal muscle or the voluntary muscle. Why is it not working? Wait a sec. So by the way, guys, today we have a special class and we'll be revising the questions in that class. Okay. So make sure that you all join me on Unacademy app. Okay. And on that app in free class, we will be practicing the real questions. We'll be practicing the real questions. Is that clear? Which appear in your exam. Now, these are the types of muscles. Let me talk to you only before I hope this starts working in a while. Okay, so types of muscles, these are the three types of muscles that we see in here. The first one, the first diagram that you see, it represents the voluntary muscle, voluntary or the skeletal muscle because it is attached to the bones. And what see the labeling, it has striations. So because of the striations, they are also known as striated muscles. Is that clear? Second labeling that you see in the first diagram is of nucleus. And see, your skeletal muscle is multinucleated. It has many nuclei on the periphery. Is this point clear? Right. The timings for the special class would be 9. I'll see you at 9 again. And then the middle diagram is of the smooth muscles. Smooth are the involuntary muscles which are present in your internal organs. Is that clear? Where are skeletal muscles present? Where are your voluntary muscles present? In all your limbs, in your tongue. All these muscles are voluntary in nature. You use them as per your own convenience and will. Right? Now see the second diagram. Smooth muscle, involuntary. These muscles are found in your internal organs. All the internal organs which are not in your control. Now see the shape. The first diagram, that is the voluntary muscle, is cylindrical in shape, while the smooth muscle is pointed at the ends, tapering ends. That's what NCERT writes, tapering ends, right? Tapering means pointed at the ends. Do you see that black dot? That represents nucleus. Now, unlike voluntary muscles, your smooth muscles will have single nucleus and that too in the center. And you don't see any striations, so therefore they are known as smooth, also known as non-striated. Are these points clear to everyone? Are these points clear to everyone? All right. The third diagram represents the cardiac muscles. So what does cardiac muscle mean? They will be present in the, yes please. Where will the cardiac muscles present? In the heart only. Only one and one place where cardiac muscles are present in heart. Are these points clear? Then there is one nucleus and you see these black colored dots. Do you see these black colored dots? Yes or no? These black colored dots, wait, let me use highlighter. Okay, yeah, this is the highlight. Yes, I want the highlighter. Okay, now do you see these striations? There are faint striations present in the cardiac muscles. There is single nucleus present, and these black bars that you see, these are the junctions present between the adjacent cells, and these junctions are known as what? What are these junctions known as, guys? These are known as intercalated discs. What do we call them? Intercalated discs. Are these points clear to everyone? Now, moving on to connective tissue. The third and the most abundant tissue is the connective tissue. So we have three types. One is the loose connective tissue, we have dense connective tissue, and we have specialized kind of connective tissue. Yes? So loose connective tissue is further of two types, adipose and areolar. Dense connective tissue is again of two types, regular and irregular, and specialized connective tissue is of four different types. So first of all, you must remember this classification. Clear to everyone? Then see this diagram. This diagram of areolar tissue is one of the most important diagrams in your NCERT. See here, areolar tissue. Just see the labelings in this areolar tissue. Now, areolar tissue is your loose connective tissue. Okay. Out of the four tissues, connective tissue is the most abundant, and out of connective tissue, areolar is the most abundant. Okay. Now, wherever there is empty space or the organs need to be packed or there is a lining that needs to be set in, areolar tissue comes in that way. Is this point clear? Areolar tissue will come in that way. So areolar tissue, see the labelings here. The red colored cells present here, these are the macrophages. These are the macrophages. Now these green colored cells, long cells, they will give rise to fibers. So they will be known as fibroblast. And these blue colored things that come out of this green cell, fibroblast, these are the collagen fibers. Is this clear? Then we have a mast cell here. So all these and these pink colored labelings, these interconnections that you see, these are the reticulate fibers, the interconnected fibers. Are these points clear? Anyone with any doubt? Okay. So what is the main function of areolar tissue? Remember, function, it forms the framework below the skin. So below your skin, which tissue is present? Areolar tissue. It forms packaging around the tissues. Is that clear? Talking about adipose tissue is meant for storing fat. Do you see there is a fat storage area? This is basically a vacuole. Then it is such a big vacuole that even nucleus has been pushed on the periphery, and then this is the plasma membrane. All those of you who feel that we have lot of fat, that fat is stored within this adipose tissue, and every one of us has this adipose tissue. We need this adipose tissue to store fat as a reserve food material. Not only that, it provides energy, it provides insulation. Okay, so these are the functions of adipose tissue. All clear? Now, this figure 7.5 that you see, this represents the dense connective tissue. So, this one is the loose connective tissue, right? While these two that you see here, these are the dense connective tissue. Now, dense connective tissue are further of two types, regular and irregular. Do you see that here there is a regular pattern in which these fibers are present? Since there is a regular pattern of arrangement, we call it dense regular. And here in the second diagram, we don't see a regular pattern. This fiber is going in this direction. This one has the mood of this direction. This is in another some mood. So there is irregular pattern of arrangement. So one with the regular pattern is known as regular. One with the irregular pattern is known as irregular. Are these points clear to everyone? Please let me know. Clear to all? Can we move on to the next then? And these diagrams represent your specialized connective tissue. What is specialized connective tissue? One is, see, specialized connective tissue is represented by your bone. This refuses to work still. Okay, never mind. Just cooperate, guys. So this is your cartilage. Sorry. This is your cartilage. This is the bone. These are the blood cells, right? So see cartilage. The cartilage forming cell is known as chondrocyte. So bone forming cells, these small insect-like structures that you see, these are all bone forming cells and we call them osteocytes. We call them osteocytes. Clear? And these are the different blood cells, RBCs, WBCs and platelets. So all of these represent what? Specialized connective tissue. All of these represent the specialized connective tissue. Are these points clear to everyone? And guys, with this, we move on to the last tissue, that is the nervous tissue. Now, nervous tissue, the unit of nervous tissue is a neuron, right? So, this is the diagram of neuron given in your book. See, The central portion, this is the nucleus, and the part around the nucleus from here to here is called cell body. Then these branches, which will bring the stimulus, these branches are known as dendrites. And then this elongated structure is known as axon. Is that clear? So the three parts of neuron are, yes please, dendrites, cell body and axon. What is the function of dendrites? They bring the stimulus. Cell body will convert it into the action potential, and axon is going to carry that away towards the next neuron. Are these points clear? Perfect. Now, in this tissue, there are two types of cells formed, guys. There are two types of cells. Just a second. There are two types of cells present in the nervous tissue. One is the neuron, and the second type of cells are known as neuroglial cells. Yes, one is neuron that you see here, but there is another type of cells also known as neuroglial cells. Now, what is the function of neuroglial cells? So, neuroglial cells are actually going to support the neurons. They're actually going to heal the neurons, so uh, repair, healing, protection, right? Everything is provided by these neuroglial cells, and they actually form 2/3 of the total nervous tissue. So 1/3 are neurons and 2/3 are the neuroglial cells. Are these points clear? So these form 70% of the total, 2/3 or almost 70% of the total nervous tissue. Is this point clear? Hanji, are these points clear to everyone? Once again, guys, those of you who have missed this information, 19th of April 2 to 5, we have a mock test series. Now, please let me know that all these two chapters are super clear to you. And now we will be revising anatomy. See, for anatomy, we have so many tables. We'll be quickly revising the anatomy. So I've made so many, yes, I've already made so many flowcharts. So let's talk about anatomy. Yes, please. Class will be till 8 o'clock. 5 to 8. 3 chapters, at least 8 o'clock. Okay? Alright, guys. Now give me a thumbs up. All of you, quickly give me a thumbs up that you are making these quick notes along with me and you are able to revise everything. Quickly. You are able to revise everything. Yes, please. Green hearts. I actually love green hearts. You know, this is so interesting. This color is beautiful. All clear? Perfect. Moving on to the next, guys. Old NCERT concepts covered in anatomy. Yes, there are. All the topics will be covered here. I have already been covering the the concepts from the old NCERT, which are in syllabus. So they will be covered here also. Okay. So anatomy as per the old NCERT is being discussed. Perfect. Now you know that I love green hearts. Now all of it, everybody is sending me green hearts. Cool. Send me green hearts. Red B? Okay. But green is new. Green is different. Green is nicer. Anyways, coming on to anatomy. So anatomy means study of internal tissues, where we are going to talk about the tissues and the arrangement of tissues. That is the tissue system. So what exactly is tissue? And the study of tissue is known as histology. So see, the definition of tissue is the group of cells similar in structure, common in origin, and have same function. These and share the common interstitial fluid. This group of cells is known as tissue. Clear to all? Now, two types of tissues are found. One is the meristematic tissue. Another type of tissue in plants is permanent tissue. Meristem, we already know, these are the dividing cells. Since the cells are dividing, they are not specialized, they are undifferentiated, right? While the permanent cells cannot divide and they are mature, they are differentiated, they are specialized to perform special function. Is this clear? Now, please see. The meristem, which is formed for the first time, is known as primary meristem. Now, listen very carefully, guys. Thank you so much for the green hearts, but now please concentrate. I will really love if you send me a green heart with your report card, with your result saying that you have cleared your NEET exam. Then send me a green heart. I will love that green heart. Is that clear? Green for biology. Yes, green for botany. Green for botany now. Thank you. Okay, guys. So the meristem which is formed for the first time is known as the primary meristem. Okay. Gradually, see these arrows. Gradually, this meristem only will change into, it will get specialized, it will differentiate to form permanent tissue. And as per the knowledge of cytology, we must know that when a cell differentiates, it enters which stage? G0 stage, right? So these meristematic cells will differentiate, enter into the G0 state and become permanent. But if required, the permanent cells can again become meristematic by the process of de-differentiation. By the process of de-differentiation. Is this clear? Now, this meristem which is formed as a result of de-differentiation is known as secondary meristem. Are these points clear? Primary means formed for the first time. Secondary means formed after de-differentiation. Is this clear? And again, this meristem can differentiate, so it will be known as redifferentiation. And after redifferentiation, the permanent tissue which is formed is known as secondary tissue. Please tell me that these three terms are clear to all of you. These three terms are clear to all of you. Yes, please. These three terms are clear to all of you. Shall we move on? Now, talking about types of meristematic tissue. See, we have primary meristem, we have secondary meristem. Now, this is on the basis of origin. Very important. On the basis of origin, we have two types of meristem. I just told you. Present since origin will be known as primary, like apical meristem and intercalary, which will be formed later from dedifferentiation is known as secondary. Are these points clear? All right. So the examples of secondary meristem are lateral meristem and interfascicular cambium, vascular cambium, or the cork cambium. So three examples for secondary meristem are lateral, interfascicular vascular cambium, and cork cambium. Now, on the basis of location, that where are they present, we have again three types of meristem. The one present at the tip of the shoot or the tip of the root will be known as apical meristem. We have root apical meristem, we have shoot apical meristem. They will help in elongation, right? Then ones which are present at the node region, they are known as intercalary. So at the node, we have intercalary, and from intercalary will arise the mature structures like leaf, tendril, and branch. And then one on the lateral side, on the side, cylindrical, in the cylindrical form, right? These cylindrical form of meristem on the sides of the plant is known as lateral. So they will help in increasing the girth of the stem. Are these points clear? Pratik, you are funny. You're very funny. You will crack everything for me. Don't crack the glasses and the bottles of your house. Don't do that, okay? Your mom will crack you then. Okay. Now, moving on to the next type of tissue. Meristem we have talked about. I hope everything is clear about meristem tissue. Now discussing the permanent tissue. Now this is well differentiated. It has a specific role to play. Now, if the permanent tissue consists of similar type of cells, we'll call it simple tissue. If it will consist of different types of cells, but same origin and overall same function, it will be known as complex tissue. Now, please just take a breath. Breathe and please understand. Calm down, breathe and understand. See, as per the definition, we say that tissue consists of similar types of cells. But complex tissue will not consist of similar type of cells. It will have different cells, right? Then why do we still call it a tissue when it has different types of cells? Reason being, the origin of all the cells is same, and ultimately all of them, they are going to perform the same function. Is this clear? For example, in complex we have two types, xylem and phloem. Now all the various elements of xylem will help in water and mineral conduction, and all the elements of phloem will ultimately help in food conduction. Okay. Now we have simple tissues, parenchyma majorly for storage, collenchyma for flexibility and mechanical support, and sclerenchyma for rigidity. Is this clear? The main function of parenchyma is storage. See, The main function of parenchyma is storage. Now, this is the most abundant tissue in the plant. Like animals have most abundant tissue as connective tissue, for plants, the most abundant tissue is parenchyma. Then, isodiametric cells can be spherical, round, oval, polygonal, or elongated. They have thin walls made up of cellulose, and they are closely packed cells with few intercellular spaces. Are these points clear? Clear? So what are the functions of parenchyma? See, this one slide will have everything about parenchyma which is written in NCERT. So main function, as I said, is storage of food. But other than storage, it is also involved in secretion, absorption, photosynthesis, providing buoyancy and sometimes support as well. Now there are two modifications of parenchyma that are in our syllabus. If parenchyma starts storing air, then we call it aerenchyma. Easy. What is this air going to help in? This air will keep the cells buoyant. It will help to float. So the plants which are going to float in water, like lotus, like water lily, they have lot of parenchyma. They have lot of parenchyma. Ishornia, they have lot of parenchyma. Is that clear? Then, if the parenchyma stores chlorophyll, we'll call it chlorenchyma. And what will that function as? Chlorenchyma will help in photosynthesis. So the palisade mesophyll layer of leaves is the example of chlorenchyma. Are these points clear? Are these points clear to all? Okay. We are going to complete this in another 15 minutes. This chapter will be done because everything is in the flowchart format. Okay. Yes. So collenchyma, the cells can be oval, spherical, polygonal. They are present below the epidermis and they usually form continuous layer or they can be found in patches. They have thick depositions of pectin, cellulose, hemicellulose. Very important. Like the cell wall of parenchyma is only made up of what? Only made up of, yes please, only made up of cellulose. In collenchyma, other than cellulose, we have pectin and hemicellulose as well. The function of collenchyma is to provide flexibility and mechanical support. Where do we see flexibility and support in the plant? In the midrib of leaf, in the petiole of the leaf. These are two very important locations, right? Then coming on to the third type of simple tissue, it is sclerenchyma. Oval, it can be spherical, they are long or they can be narrow, but the most important point is sclerenchyma has special deposition of lignin in the cell wall. Now this lignin will make the cell hard, it will make the cell rigid. In fact, this lignin deposition can increase so much that see, the entire space will be filled with lignin. There will be no space left for the protoplasm. So loss of protoplasm will kill the cell. The cell will die. So many sclerenchymatous cells eventually become dead cells. But then dead cells play important role in protection because there is no fear of death anymore. They are hard. They provide rigidity. They provide mechanical strength. Are these points clear to all? Please let me know. Now see, if this sclerenchymatous cell is round, oval, and or spherical and has thickened cell wall and a narrow cavity like this, we call it stone cell or sclereid. Is this clear? But if the sclerenchymatous cell is long and elongated like this, then we call it a fiber. So on the basis of shape, we call it either stone cell or we call it fiber. See the examples of stone cells. Where do you see this sclerenchyma? In the fruit walls of nuts. That hardness of the nut shell is because of sclerenchyma. Pulp of fruits like when you eat guava or sapota, sapota is chiku, or you eat pear. So these fruits have that gritty feeling when you are eating that. That grit, that gritty feeling in the pulp is basically the stone cell. Are these points clear? And then seed coats of legumes and tea leaves, all these have sclerenchyma. Clear? Moving on to xylem, the complex tissue. See, commonly this xylem is known as wood, mainly dead tissue, conducts water and minerals. The direction is always from bottom to top. That is unidirectional. So it helps in conduction and also provides mechanical strength. There are four elements of xylem. One is xylem vessels. Then we have tracheids, we have fibers, and we have parenchyma. Now, overall, xylem is called a dead tissue because out of these four elements, only one element, which is parenchyma, is the living one. Rest, see, this is dead, vessel is dead, trachea is dead, and fiber is always dead. Why they are dead? Because they are made up of sclerenchyma, sclerenchyma, sclerenchyma. Do you see all these points here? Do you see all these points here? Now, vessels are only found in angiosperms. That's how they get their name. Angiosperms. Angiosperms means seed-bearing plants which have angion means vessels in their xylem. Is that clear? Then tracheids are pointed and tapering. So in our old NCERT, there were two diagrams and this diagram was in your PYQ. Identify this diagram. That was a question. So remember, this cylindrical structure is called a vessel while tracheids are pointed. Tracheids have tapering ends. So the tapering one which is pointed is the tracheid. T for tapering, T for tracheid. Can you remember it like that? T for tapering and T for tracheids. Cool. And these cylindrical ones, these are known as vessels. Will you be able to identify now? Give me a thumbs up that you will not make any mistake in identifying these two diagrams. Yes, please. How many of you are going to be very, very sure about these two diagrams? Clear? And then moving on to fibers. Fibers, as we have read, they are always long and pointed. Obliterated lumen, because obliterated means disappeared lumen. Why? Because the whole space is filled with lignin. They might have cross walls like septate or they can be aseptate also. Perfect? Perfect. And then parenchyma is always living. Now see here, one thing I want to share with you. See here, see here, see here. Now see here. So xylem parenchyma. See, xylem parenchyma is the only living element in xylem. Okay, they are living, and xylem is made. Sorry, parenchyma is meant for storage. So what does this xylem parenchyma stores? Remember, it stores tannin. What does it store? It stores tannin, food, and water. Is that clear? You will get these PDFs on the Telegram channel. Now, coming on to phloem. So, phloem also has four components. Food conducting tissue, it is mostly living. Sorry, it is mostly living, and there are bidirectional movements of food, upward also and downward also. Now, mostly living because only these phloem fibers are dead. Rest all the three components are living. Let's read. Parenchyma will store food and other substances like renin, latex, resins, latex, and mucilage. See, remember it. Permal. Remember it as thing we have presumed. Is that clear now? Connected. Okay. So epidermal tissue system, it is the outermost tissue system. It is protective in nature. Then it is meant for absorption and secretion, right, and prevents transpiration. See what all comes under this tissue. The single-celled epidermis, which is always made up of parenchyma. Usually, epidermis is single-layered, but only in xerophytes, only in desert plants, it can be multi-layered to prevent transpiration, right? Then the second structure which comes under epidermal tissue system is epidermal appendages. Like roots have root hairs and stem has stem hair known as trichomes. Root hairs are always unicellular. Stem hairs can be unicellular or multicellular. Root hairs are never branched. See here, root hairs are never branched. They are always unbranched. While the stem hair, I've drawn them together, stem hair can be branched and they can be multicellular also. Are these points clear? And then third component of the system is cuticle. So cuticle is present in stem as well as leaves. So young stem will have cuticle when it is green, and the leaves always have cuticle to prevent transpiration. Then the fourth component is a stomatal apparatus. Are these points clear? So what does stomatal apparatus consist of? Stomatal apparatus has three components. Remember, guard cells, stomata, and subsidiary cells. Clear? So monocots will always have dumbbell-shaped guard cells and dicots will have bean-shaped guard cells. Clear to everyone? Now, coming on to vascular tissue system. Now, remember these terms. Please see. Whenever the cambium is present, see, this is the vascular bundle. If the vascular bundle has cambium in between, it makes it open vascular bundle. If there is cambium, it is open. If there is no cambium, it is just like this. Okay, so there is there is phloem and this is xylem. In between xylem and phloem, if the cambium ring is present, cambium band is present, it is open. If there is no band of cambium, it is closed. So as a matter of fact, dicots will always have open vascular bundle and monocots will always have closed vascular bundle. So how do we remember? DOC. Dicots have open vascular bundle because there is cambium. Is this clear to all? Will you be able to remember that? And then on the basis of, now see, on the basis of arrangement of xylem and phloem, if xylem and phloem are present in alternate patches like this, we call this as radial arrangement. Radial arrangement is always seen in roots. So for our NEET preparation, we have to remember RR. There is this movie RRR. We have to remember double R. So roots have radial arrangement. And what is radial? So xylem and phloem are present on the different radius. In case of conjoint, see, xylem and phloem are present on the same radius, in the same bundle. Is this clear? So conjoint is seen in stem and in leaf. Now on the basis of location of protoxylem, see, if protoxylem is on the outer side, this is the outer side, right? If protoxylem is on the outer side, this condition is known as exarch. If protoxylem, this is the protoxylem is on the inner side, then this is known as the endarch. Stem and leaf will always have endarch and root will always have exarch. Are these points clear? So three things that you always see in roots. Roots have radial arrangement, roots have exarch condition. Is that clear? Is this point clear? All right. So moving on to now this one table. This one table will summarize everything for you. See, epidermis in case of dicot and monocot root. So first of all, epidermis in all roots and leaves are single layered and they are always made up of parenchyma. You must take a screenshot for this and you will always obviously find it in the notes also. See here. See here. Open means it is open. Open means since cambium is present, it is open to secondary growth. Secondary growth can only happen if there is cambium, right? So open means open to secondary growth. Now see, this table will revise everything. Epidermis is present in all root, stem and leaf. Okay, I'm just comparing root and leaf, stem here. It is always single layered except in case of desert plants. Made up of parenchyma in all. Clear? Now in case of root, be it dicot or monocot, there can be root hairs present for water absorption. They are unicellular or unbranched. While in stems, epidermis can form stem hair known as trichomes. They can be uni or multicellular, branched or unbranched, and they will have cuticle as a waxy coating to decrease the transpiration. See, cuticle will never be present in root. Here we don't have cuticle because if root will have cuticle, how will it absorb water? Because cuticle is a waxy coating, right? It will prevent water absorption. So roots will never have cuticle. Is this point clear? No, next class will be on Saturday. Yes, tomorrow we do have a class. Yes. It's Friday. Tomorrow, guys, we are going to revise the entire 11th biology. Okay? Yeah. Now, talking about cortex. So, cortex in case of root is multi-layered, made up of parenchyma and intercellular spaces are present in both dicot as well as monocot. Clear? But then next to cortex, the innermost layer of cortex is always known as endodermis. So innermost layer of cortex is endodermis and in root, be it dicot or monocot, they form special strips known as Casparian strips. Casparian is in the name of the scientist. So basically, there is specialized suberin deposition in Casparian strips. Now suberin is water impermeable. So endodermis cells which have Casparian strips don't allow water transport. So in that case, we have some passage cells. They have suberin deposition and there are barrel shaped cells. Is this clear? Now, inner to endodermis is pericycle. Again, it is few layered, made up of parenchyma. And what is the function of pericycle? It is this pericycle which gives rise to lateral roots and vascular cambium. Is this clear? It gives rise to lateral roots and vascular cambium. Inner to pericycle is pith. Pith is always made up of parenchyma, be it root or be it stem. Are these points clear? Okay, now see the vascular bundle. In case of dicot root, now see here very carefully. In case of dicot root, guys, the vascular bundle, being a root, it is radial and exarch. Now dicot will have open vascular bundle, cambium will be present, but in monocot root, again it will be radial, again it will be exarch, but it will be closed vascular bundle, means the cambium will not be present. Are these points clear? Now moving on to dicot stem. Again, single layered epidermis made up of parenchyma, trichomes present, cuticle present. Talking about the dicot stem, listen very carefully. Now, dicot stem has cortex divided into three regions: hypodermis, cortical layers, and endodermis. Are these points clear? Are these points clear to all? The picture is super clear, I feel. Why are you just disturbing? See, I don't think you will find such a beautiful flowchart anywhere. Honestly, it took me a lot of time. And I feel everything is summarized. You don't need to read NCERT beyond this, after this. Just see the diagrams, that's it. See, cortex of dicot stem is made up of three layers. It divided into three layers: hypodermis, cortical layer, and endodermis. Now, hypodermis made up of collenchyma, cortical layers made up of parenchyma, and endodermis has starch grains. So, endodermis here, which is the innermost layer of cortex, is also known as starch sheath and made up of parenchyma. Okay. While in monocot stem, see, the cortex is not very well differentiated. We only call it hypodermis, made up of sclerenchyma, and rest of the whole tissue is known as ground tissue. Clear? Ground tissue is made up of parenchyma. Now talking about pericycle. See, in root pericycle is made up of parenchyma, but in dicot stem it is made up of sclerenchyma, and in monocot it is just absent. Talking about pith, I told you pith, no matter where it is present, it is always made up of parenchyma. In dicot root, in monocot root, in dicot stem, parenchyma. While monocot stem will not have pith at all. The entire space is filled with ground tissue. Are these points clear? Are these points clear? Let me fix it here. Now see the vascular bundle. The vascular bundle here, wait. The vascular bundle here is made up of, see, vascular bundle of dicot stem has conjoint arrangement. It is dicot, so it is open. It has cambium ring, and the vascular bundles are arranged in the ring arrangement. They are in the ring arrangement. While in monocot, they are closed, they are conjoined and endark, but closed, and they are scattered. Are these points clear to all? Please let me know that these points are absolutely clear to all of you. And last not the least, guys, vascular bundle. See, there is Krenz anatomy. What is Krenz anatomy? When there are specialized bundle sheath cells present around the vascular bundles, we call it Krenz anatomy. So bundle sheath means these sheaths, they form a covering around the bundle sheath cells. They are always made of sclerenchyma. They are found only and mostly in the monocots, but at few times they are also seen in dicots. Is this clear? And Krenz anatomy is specialized for C4 cycle. Yes or no? They are specialized for C4 cycle. Clear to everyone? And then last not the least, we'll talk about bulliform cells. See, bulliform cells are only in the monocot leaf along the midrib. See, if this is the leaf of the monocot, this is the midrib, and around the midrib are present these swollen cells. These cells are called bulliform cells. Now, when they have water, when the water is present in these cells, the leaf will be exposed, right? When the water is lost from the cells, under water stress, the leaves will curl inwards. So these are specialized cells. When the cells they have water, they are turgid, the leaf surface is extended. And when there is loss of water, it leads to curling of leaves, not outwards but inwards, to reduce the surface area and check transpiration. please let me know that these points are super clear to all. And guys, with this, we complete our unit-wise final revision. I'll again tell you that families, cockroach and frog, they will be revised in a separate session, 15-minute session of cockroach and frog and of families. Okay. So thank you so much for joining me, and please join me again in the app, Unacademy app, for the practice session of questions based on Unit 1. The previous chapter that unit that we have revised, today I'm going to practice the questions for that. Is that clear? Telegram channel, the link is given in the description box, Prateek. Please join the channel. Join the channel. Otherwise, you can find out, in case if you want to hunt it yourself, then you can find out this Unacademy NEET English channel on Telegram. You can easily find it and find the notes as well. Is this clear? Thank you so much, guys. I'll see you again tomorrow, because tomorrow is a big day. It's going to be a marathon session starting from 7 p.m. and we are going to revise the entire 11th biology NCERT with PYQs. With PYQs. Okay. Thank you so much. Thank you so much. Okay, keep revising, and I'll see you at 9 on the app in special classes. Bye bye.